Okay. So they have given you a reaction. They are going to ask you a question. A biochemical shunt reaction is shown below. Identify the incorrect statement. Identify the incorrect statement. This reaction occurs exclusively in erythrocytes. Okay, that is the correct answer. Increased 2,3-BPG will reduce oxygen affinity for hemoglobin. Yes, that is also because it releases or kicks off O2, unloads oxygen. Both the reactions are catalyzed by 2,3-BPG mutase. That is also correct. Mutase is the one which catalyzes both. One ATP is synthesized in conversion of 2,3-BPG to 3-PG. No, it is actually 2,3-DPG to 3-PG. One ATP is not synthesized. Rather, one ATP is released. So this particular cycle is called as Rappaport-Lubering cycle. See, it is a detour from. So Rappaport-Lubering cycle is a detour from glycolysis. It happens only in RBCs. Production of 2,3-BPG. Okay. And the role of 2,3-BPG is it is a oxygen unloader. It is a oxygen unloader. It releases O2 from hemoglobin to tissues. And when glycolysis proceeds via Rappaport-Lubering cycle, the net yield is 0. Why the net yield is 0? See here, usually 2 ATPs are consumed in the first in hexokinase and PFK1 reaction, and then 4 ATPs are produced in substrate level phosphorylations. Okay, so net yield will be +2. Net yield will be +2. Okay, and then along with NADH it will be 7 ATPs. But when it goes via Rappaport-Lubering cycle, in this 4 ATPs, 2 ATPs are not released. 2 ATPs are not used because there is no mitochondria in RBCs. There is no mitochondria in RBCs. So what will happen? Totally 2 ATPs are produced, 2 ATPs are consumed. The net yield if the glycolysis proceeds via Rappaport-Lubering cycle is 0. That is a 0. So RBCs usually itself only there is no mitochondria. So all the glycolysis will be what type of glycolysis? It is anaerobic glycolysis which produces lactate. So in RBCs it is anaerobic glycolysis produces lactate. Okay, so that will be the answer for this question. The answer will be 4. A biochemical reaction is shown below. Identify the wrong statement. Which is the wrong statement? What is this? Okay, this is from the hexose monophosphate shunt. This reaction is part of the HMP shunt, correct? This reaction is catalyzed by transketolase is correct. This reaction occurs in the cytoplasm is correct. NADP is cofactor in this reaction is wrong. Why? See, HMP shunt produces NADPH. But then where it produces? NADPH is produced in the first and third step of HMP shunt. That is G6PD step and phosphogluconate dehydrogenase step. In these two steps only NADPH is produced. After that, it enters into the second phase of the HMP shunt where the recombination occurs. You all know the recombination, right? So we have a 15 carbon compound. It will be split into 5 plus 5 plus 5. Okay. And also with production of ribose 5 phosphate. That will be going for purine synthesis. Now we will take this 5 plus 5 and convert them into 7 plus 3. This 7 plus 3 will be converted into 4 plus 6. Keep this. This 4 and this 5 will be converted into 6 plus 3. I can write the names also. Fructose 6 phosphate, glyceraldehyde 3 phosphate, again fructose 6 phosphate. So this is the whole thing. They are asking you about this step. They are asking you about this step. So transketolase, transaldolase, transketolase. That's all. TK, TA, TK. This transketolase is the marker for vitamin B1 deficiency. Please remember that. That is an MCQ. Transketolase is a marker for vitamin B1 deficiency. Okay. So that will be about a HMP shunt. Okay. So this is a very easy question. Okay. Next question. Entry points of amino acids in TCA cycle to produce glucose by gluconeogenesis are shown below. Identify the incorrectly matched options. 1, which is 1A. Okay. So pyruvate, what they are? Pyruvate comes from serine. Okay. Next one. B. Sorry. Phenylalanine. Phenylalanine fumarate. So they are saying serine, phenylalanine. 3. Valine. Succinyl-CoA. Valine. Alpha ketoglutarate, cysteine. So which is wrong they are asking. See, the wrong is cysteine alpha ketoglutarate. Why? Because I am going to teach you. See, pyruvate can come from mainly serine and alanine. It can also come from hydroxyproline. Okay. All those things. But then serine can give rise to pyruvate. Okay. So that will be the correct answer. And then fumarate. Fumarate will come from phenylalanine and tyrosine. So that is also a correct answer. Succinyl-CoA will come from, what is a mnemonic? Vitamin M. Valine, isoleucine, threonine, methionine. Valine, isoleucine, threonine and methionine. That is about succinyl-CoA. Alpha ketoglutarate mainly comes from glutamate. This is the crux reaction of transamination. It does not come from cysteine. It does not come from cysteine. And the other one you should remember is oxaloacetate comes from aspartate. This is the other transamination reaction. And for all transamination reactions, you need a vitamin B6, pyridoxal phosphate. And this vitamin B6 pyridoxal phosphate is also needed for glycogenolysis. That is a very often asked question in JIPMER exam. But nowadays it is not JIPMER exam. So B6 is needed for, PLP is needed for glycogenolysis. And the B6 or PLP is also needed for heme synthesis first step. That also you must remember. So that is about this thing.